Hello and welcome to this Australian Biocommons webinar. My name is Melissa Burke. I'm the Australian Biocommons Training Manager and I'll be your host for today. This webinar is part of a series in which we share useful information about the latest digital techniques, data and tools that are available for the life sciences community. Each month we hear from local and international experts who present on a bioinformatics topic that will help Australians to deliver their best environmental, agricultural and medical research. You can keep up to date with the latest news and events from the Biocommons via the links that you can see on your screen. Before we begin, I'd like to take a moment to acknowledge the traditional owners and their custodianship of the lands on which we meet today. Today, we're joining you from the lands of the Turrbal and Yagara people, the Gadigal people and the Wurundjeri people. We pay our respects to their ancestors and their descendants who continue cultural and spiritual connections to country, and we recognise their valuable contributions to Australian and global society. Today, we're thrilled to welcome Dr. Hossein Valipour Kahrood and Dr. Cali Willet as our speakers. Hossein is a bioinformatician at the Melbourne Proteomics and Metabolomics platform, where he enjoys supporting a variety of different research projects. He has a diverse array of experience, including Shiny app development, cancer genomics, and linear modelling of genomic selection in breeding. Cali is a senior research bioinformatician at the Sydney Informatics Hub, where she supports researchers to use HPC and develops open source, efficient and scalable bioinformatics workflows for high throughput datasets. Her expertise and interests span a variety of projects, including human genetics, cancer studies, livestock rare diseases, wildlife and companion animal genomics, and metagenomics, just to name a few. Today, Hossein and Cali will be introducing us to the concept of functional enrichment analysis and how you can use this to make sense of those gene and protein lists that you often get from omics experiments. Welcome to the webinar, Hossein and Cali. I'm now going to hand it over to you to get us started on this topic.
Thanks, Melissa. And welcome everyone to our today's webinar on functional enrichment analysis. This webinar is going to be an introduction to the workshop that we're going to deliver in a few weeks' time. So basically, we're going to, in this webinar, give you a background of functional enrichment analysis and some statistics behind the methods for functional enrichment analysis in part one. And also we are going to introduce some of the platforms, GUI, web-based tools, and R packages to basically run functional enrichment analysis, and also how to choose between the two. So basically today's webinar is going to cover these introductory material, which can be an introduction to our workshop, as I said, in a few weeks' time. All right, let's start the part one. In part one, I am going to give some background and a statistical concept behind the functional enrichment analysis. So what is a functional enrichment analysis? So in general, it refers to various methods which are used to extract biological or functional insights from a list of biomolecules, such as genes, proteins, metabolites, etc. And this is to identify biological functions, pathways, mechanisms that are significantly associated with a subset of biological molecules, such as those ones that are differentially expressed in a differential expression study like an RNA-Seq for a particular condition. All right. Before I start, I need to come up with some of the synonyms which have been used in the field for functional enrichment analysis. They have been used quite similarly, and they are pointing similar things. Terms like enrichment analysis, pathway analysis, pathway enrichment analysis, functional annotation analysis, or the one that we are going to use is the functional enrichment analysis, for our webinar. A functional enrichment analysis workflow, it will be something like this graph that is being presented in here in general. It starts with an omics experiment, which renders some list of features, and that feature can be, for example, a gene. And from then onward, you will take some methods to analyze your enrichment analysis. For example, you can take a list of genes which have been adjusted for their p-value or fold change as a cutoff filtering. And together with some background, you present them in some ORA analysis tools to do the enrichment analysis. Or you can take the entire list of genes and also take those without filtering those lists and make some input for gene set enrichment analysis using those tools to do the enrichment analysis. And either of these is going to use a number of databases, depending on which web-based or R tools you're using to do the enrichment analysis for you to get the enrichment result. I am going to give a very simple example of a functional enrichment analysis of a mouse diet experiment, just to understand what it's going to like from start to end. So imagine we've got an experiment where we have two groups of mice. One have been in a high-fat diet for some time, and the other one was on a low-fat diet for some time, let's say for a month or two. And after that period of time, we have been doing some sort of omics analysis like RNA-Seq, of which there have been, let's say, like 12,000 genes come up on that experiment. And we realized that according to our filtering, there have been around 40 genes have been differentially expressed between these two conditions. What we are going to do, we take those set of genes or those list of genes, and then using some of the annotation databases available online, we plug them together using web-based tools. It will calculate some sort of statistics and analysis, and it comes up with some p-values in the result, like a table. And basically, the enrichment analysis come up with some sort of result like that most of the time, apart from the visualization. You can take the result and do further analysis. For example, in here, I'm just showing a chart, a dot plot of KEGG pathways, which lists the pathways have been enriched in this experiment. In this KEGG pathway, as an example, it shows the gene ratio, or basically the number of genes in the term, or the percentage of genes in the term on the x-axis, and also their p-value. And if you can take this further, for example, you can take one of these and go to KEGG database and have a better look at what the actual KEGG pathway is. And for example, this is one of these pathways, and it shows that these genes have been impacting these two parts of the basically pathway of interest that we are interested in our research, coming up from differentiation between high-fat and low-fat diet. So this is an overall overview of a functional enrichment analysis. All right, so question is, when we do the functional enrichment analysis, normally we do it after a differential expression, when we have an omics study. A differential expression is like transcriptomics, proteomics, lipidomics, metabolomics, or epigenomics, and this list can go on. And in there, we have, for example, two conditions or multiple conditions. Let's say high-fat diet versus low-fat diet, or let's say tumor tissue versus healthy tissue, etc. And when we do this omics study, they are coming with a very large scale result. They are high throughput studies. They come up with high number of features. And most of the time, the researchers don't know what to do with those big number of genes, proteins, metabolites, etc. So the idea of functional enrichment analysis is to summarize this long list of features it can be genes or proteins, and then to extract meaningful biological insight from this data. Yeah, so we are going from hundreds to millions of features to, let's say, 100 to 1,000 or so of the pathway or gene ontology or etc. at the end. We would like to know what genes behave differently between two conditions and what does this tell us about the condition of interest. And basically, the enrichment analysis can also help us to hypothesize or generate some hypotheses. For example, dysregulation of DNA repair mechanism is a key driver in tumor progression in this specific cancer type. So we can test this hypothesis through functional enrichment analysis. How we do the enrichment analysis? We basically need two main components. One is your input list, input data, and the other one is the databases. The databases are already available online, but what you need to have is the list of features, a background set, a ranked list, or gene sets for the analysis, and they are relevant to different types of analysis. I'm going to give you an example of a list of features. Here is of those 40 genes that I earlier showed. And these are the fold change of the change from high fat to low fat diet. And they're coming up with some p-value. And here, for example, let's say we have gone for p-value less than 0.05. And we usually take this alongside a background set, which I explained what it is, and then run the analysis. In another method, we take the entire set of genes, which I'm showing parts of it in here. We don't need to do a cutoff to filter out. We take the entire gene, we rank them by some sort of stats, and we use some gene sets. For example, here I'm giving you some examples of gene sets. These are hallmark gene sets. They are listed each row with different number of genes. So basically, we're going to test whether our list have been enriched in any of those gene sets. All right, there are some concepts now to consider. What is the gene list? As I've mentioned before, the gene list can be a filtered list of genes, like those coming from a differential experiment. Or it can be a list of genes which are clustered in a time point to another time point together, like those genes that you analyze in a WGCNA analysis. Or it can be the entire set of genes, not just filtered set. It can be the entire genes in your experiment. What is a gene set? Gene set is different from gene list. With the gene set, these are available online, and with the gene set, we mean like a gene ontology term or gene ontology class, a KEGG pathway, a reactome pathway, a hallmark gene set, etc. These can be our gene sets. These include different number of genes, and sometimes they overlap. Another concept to know is the p-value and false discovery rate, which is relevant both for the expression analysis step, which is before the enrichment analysis. It's where when you want to filter your list of genes, you need to know the p-value and false discovery rate of them. And also, once you have done your enrichment analysis, you come up with p-value and false discovery rate for the enrichment terms. So there are two sets of p-values and false discovery rates in this experiment, in these studies. The term regulation is basically talking about the direction where our genes are regulated. They are either upregulated or downregulated. And with the background set, it is very important to notice that background set is very critical in the functional enrichment analysis. Background set is even more important than correcting for false discovery rates. So using the right set of background is very important. You need to consider that. Let's say if we have run a liver tissue experiment and we want to find out what are the expressed genes in the diseased liver tissue versus the normal one, you do not want to take the entire human genome as the background set, because not only all of genes can be basically expressed in any any cells or any tissue. So you might come up with the set of genes in your experiment, which are basically coming from liver. So you're taking those as your background set. So you don't want to basically specify the entire genome, and it will give you some false positive or false negatives. With the ID mapping, you need some software, which needs, for example, Antares IDs instead of ensemble IDs, or etc. There are tools that can convert one ID to another. And the annotation database are important. There are a number of annotation databases available. Some of the web tools use some of them, others use other, and some can use a number of them like gprofiler. Using annotation database is very important in that what to choose, using what tool, because it can impact your result. So let's talk about the types of enrichment analysis. They have been discussed in this study, which is 10 years of pathway analysis. In 2012, it is still valid. It's been listed like gene list based as first generation, and rank based second generation, and network based as third generation. What are those? In that paper, they specify it in this way. With the overrepresentation analysis, we've got a list of genes, let's say differentially expressed genes, and we take that alongside with a background set, and we plug them into some web tools plus the database and analyze the data. With the functional class scoring or ranked-based list, we have the entire set of lists. We rank them based on some sorts of stats, and then we analyze them with relevant tools. And in the third one, we've got the pathway topology-based enrichment analysis, where we've got either a gene list or the entire list, and alongside with this topology, which has some information in it, like number of reactions, position of the genes in the pathology, types of reaction, etc., and it come up with pathway impact factor. So these three types of analysis have different usages, but most of the time people are needed to use all of them to cross-check the result. With overrepresentation analysis, as I said, apart from using your differentially expressed genes or proteins or metabolites, you can use a cluster of genes which is coming from WGCNA, or it can be a set of genes which are specific to a cell type. With gene set enrichment analysis, if you are using the desktop platform from MSeqDB, you just provide your expression data, but otherwise a lot of tools just need the pre-ranked gene list of genes in your experiment and run them from there. There is one concept called single sample gene set enrichment analysis, which doesn't look at the phenotype or the labels. Let's say we have two classes of condition or two conditions. It doesn't look at those. It just looks at the samples and does the gene set enrichment for each samples. I would like to say that in our workshop and webinar, we are not going to touch single sample gene set enrichment analysis or topology-based pathways, but there are tools that you can do it. The reason that I'm not going to use the TPA is that it requires good understanding of network biology and pathway topology, and it needs high quality pathway topology information, which may not be always available across all organisms or conditions. And that most of the time in the literature, ORA and GSA have been used. So these are most tangible examples that we can use. Question might be for you, when and why we use ORA or GSA. I've provided here a summary table of features that can tell us the different characteristics of those. With the ORAs, as I said, the input can be a differentially expressed genes or gene modules, whereas GSCA have a full ranked gene list or the entire set. With the ORA, we need to come up with some sort of cutoff using either log-off or p-value or both. GSA doesn't need cutoff. A statistical method behind ORA are Fisher exact test and hypergeometric test. GSCA is using permutation type test. The output from ORA is basically enriched term with p-value and FDR. In GSCA, we come up with enrichment score and p-value and FDR, and normalized enrichment score. What are the pros and cons? ORA is simple, computationally efficient, easy to interpret the results, whereas GSCA has some other pros, like it capturing the subtle changes in your coordinated trends across all features. So you have the entire list of genes, and then it will take the small information from one gene to another and the relationship between them. What are the cons? With ORA, it misses these subtle and yet biologically important patterns, and it assumes gene independence. It doesn't know that genes are actually related to each other and can impact each other. Whereas in gene set enrichment analysis, the cons can be ranking your list, depends on what ranking you're using. You might bias your result, and it is biased by the gene set size. The bigger the size of the gene set, it can be more enriched, so we may have false positives. There are tools that can normalize for this set, this size, and that it has complex statistical framework, and it can be computationally intensive depending on the permutation number that you're setting. If you're going away from, let's say, 1,000 to 10,000 or 100,000 permutation, it can take a long time. So let's a little talk about the statistics overview of ORA. So ORA, we are basically, what we're going to do is to test whether the number of genes that are in our differential express gene list from our experiment, let's say RNA-Seq, and overlaps with a term of interest. Yeah, it can be a pathway, it can be gene ontology, etc. We want to see whether this overlap is statistically significant or not, for which we create a contingency table where you have like the number of, in here I have 150 list of differentially expressed genes, 50 of them are in the term, 100 of them are not. The term size is 250, and 200 of them are not in the gene list. So we are basically going to test this overlap using these two lists plus the background. For this, there are a number of tests like Fisher exact test, which was introduced by Ronald Fisher. And I'm not going to talk about the history behind that. So, and that is the formula. If you run the formula for this, you come up with a significance value. Another test is hypergeometric test, and that was coming at the time when we were talking about probability theory. And this is a formula for hypergeometric test. And it is mentioned that for more precise and for a small sample size, you would go for Fisher exact test, whereas for a larger number sample size, you go for hypergeometric test or chi-square test. All right, regarding gene set enrichment analysis, what are the statistics behind it? So with the gene set enrichment analysis, I'm gonna explain it through this graph, which was published in this paper here in 2005, around 20 years ago. So imagine you've got two conditions, A and B, and once you have run your experiment, you come up with the entire list of genes in your experiment. And some of them are upregulated in, let's say, condition A. Some of them are downregulated in condition A and vice versa. So you use some stats to list and rank all of these genes. And what you're going to do is to take this list and test them against some gene sets, as I said earlier, which are available online, like a gene ontology, a pathway, etc. So what it does, it looks at this gene list, which is ordered, and see if there was a match in the set. It increases increments the value in its random walk by a value called running sum by this amount. And if there was no match between the list and the gene set, then it will decrease the value by a running sum. The running sum for increment is higher than the running sum for decrement. So it's like if there was a match, it was going to give it the reward, so it was higher running sum. If there was no match, it doesn't give it the reward, but it doesn't penalize it too much. So it goes all the way through the entire list, and we come up with a graph where there will be somewhere in our graph where we have the maximum deviation from 0, and that is our enrichment score. And this enrichment score depends on the size of the gene set, which needs to be normalized. That is why we have this normalization enrichment score in here in this step. But once we have calculated enrichment score, we need to do some calculations to get the p-value for this gene set enrichment analysis. And this is done by randomly assigning phenotype labels through permutations. Say, if you have 10 samples for 2 conditions, 5 from each, the maximum permutation that you would do if you've done some sort of binomial coefficient calculations, there will be like 252 permutations. But the number of samples is higher. If, for example, if there are 20 samples, then the maximum permutation can go really high, like 10,000 or 180,000 or very large. So you need to set a permutation number that can be reasonable for your calculation time. So with this permutation, what it does, it randomly assigns phenotype labels to samples as if originally they were not labeled, reorder the genes, recompute the ES for each gene set to generate a null distribution like this for both negative enrichment score and positive enrichment score. These are all theoretical this null distribution of p-values. And if we have our gene set enrichment, if we have the enrichment score value in here is around 0.45, and it is positively regulated, or I should say it is on the positive side with positive score, we look at the positive side of the null distribution, and it says that, oh, if I look at the graph, it shows that around 60 to 70 times out of 1,000 times, I actually had an enrichment score of 0.4 or 0.405, or 0.5 or greater than that. And if you do some calculation, it comes up with a nominal p-value of 0.09 or so, which means that this enrichment score is not significant according to our null distribution. But in the right graph, you can see we've got an enrichment score which is negative, and the enrichment score again is around 0.35 or so. And if you look at the negative side, you can see 0.35 or so, it lies in here, which means there will be a very few times out of 1,000 times that we had enrichment score of minus 0.35 or less than that. So it tells me that it is extremely statistically significant, even with the FDR p-value. So I hope this explanation makes sense, and I think I'm running a bit late with the time. Melissa, can you tell me how much time do you have?
We are at 1/2 past.
So
there is about 20 minutes left.
Oh, there's 20 minutes left for me?
For the whole thing.
Oh, so I've got 5 minutes. Okay. So that's how this p-value is calculated for gene set enrichment analysis. This gene set, as I said, it can be a gene ontology, it can be pathway like mitochondria, map kinase signaling pathway, cell types, cell cycle control, etc. So once we've done all these 3 steps, we need to also do a multiple hypothesis testing at the end. It can be FDR or family-wise error rate. This is native to MSigDB, and they, even in their paper with their data set, they couldn't find any gene set of statistical significance because family-wise error rate is very stringent and it doesn't usually come up with significance level. And so they are recommending also use FDR as a false discovery rate for multiple hypothesis testing. All right, so I've been talking about annotation databases and pathways and gene ontology, etc. I'm going to go a very quick overview of those. So these are some of the most important annotation databases like gene ontology, cake, reactome, MSeq, DB, etc. And in the upper part of my list, you can see mostly databases for pathways and gene ontology, whereas in the bottom part of the list, you can see databases for proteins, for protein interactions, for protein domains, and etc. I'm gonna skip this table and talk a little bit about what pathways are in biology. Biological pathways, like those that you can find under cake or reactome or else, are a series of interconnected biochemical reactions or molecular events that occur within cells, tissues, organs, or entire organisms. These pathways describe the flow of biological information, matter, or energy that leads to specific biological outcomes. So different types of pathways are categorized like metabolic pathways, genetic pathways, signal transduction pathway, immune response pathways, cell cycle pathways, which you can see them like these on the cake database. This is, for example, metabolic pathways. There's a lot of signals that you can see from one to another. These one are genetic pathways, etc. With the gene ontology, what gene ontology is a gene ontology is a controlled and structured hierarchical vocabulary for describing the properties and functions of gene products. With the hierarchical parents and child terms, it means the more general processes are towards parents and more specific descriptors are toward child. And with gene ontology, we've got three main domains. And I need to mention that this gene ontology was developed in 2000, 25 years ago, by the Gene Ontology Consortium. You can read this paper for that matter. So gene ontology, usually you can see these sorts of graph. It's called DAG, or directed acyclic graph in gene ontology analysis. And they include GO or Go classes or terms. They have unique identifier, they have a name, they have relations, and some other characters which are described in gene ontology. And we say directed because they are always from one to another, not the other way around, or they're not acyclic because they're not a cycle. It can be read from top to bottom or from bottom to top. So the root is the parent. Any terms toward the roots are parental, and toward the leaf they are like child. Then the types of relation in directed acyclic graph can be is A is part A, or can be inferred. For example, A is A B, and B is part of C. As such, we can infer A is part of C. As an example, I can give this mitochondrion. Mitochondrion is part of a cytoplasm. Yeah, it's an organelle inside cytoplasm, inside the cell, and it is a or an organelle. And we also know that organelle membrane is part of organelle. So this is how the relationship is mentioned. Let's skip this. As I mentioned, gene ontology has got three main domains. With molecular function, it is basically talking about molecular level activities performed by gene products, and normally molecular functions are entitled with activity in their names. Let's say the Go molecular functions are often, let's say we have a protein kinase, and if you want to say the Go for the kinase, it will, the molecular function Go for that will be protein kinase activity. And with the cellular component, you're talking about the locations where a component is relevant. And with the biological process, we're talking about the larger picture or the larger processes or biological programs. Let's say we are talking about DNA repair or signal transduction. These are large biological processes. I'm going to give another example of GO, let's say cytochrome C. The molecular function for that would be oxidoreductase activity. The biological process for that would be oxidative phosphorylation. And the cellular component for that would be something like mitochondrial intermembrane space. This is my last slide, I hope. Yes, and that is going to give us a little bit of introduction of evidence code by gene ontology. Basically, gene ontology includes a lot of genes and their annotations and the products, which are the proteins, but you need to know how they have been derived. Most of the time, they have been coming from experiments. So they are coming from experimental designs, which are quite valid. And sometimes they are phylogenetically inferred. And as such, they can have different codes which represent the relevant evidence code. For example, if genes coming from a direct assay, there will be like IDA as a code for that. And we can move away from here towards some other computational analysis or even statements by authors, etc. But the thing is, when you move away from top to bottom, the strength of the evidence will be less and less, because the top part is relevant to experiments that have actually been informed in the wet lab or have been some rigorous phylogenetically analysis performed. Whereas down the bottom, you have some statements or some electronic annotations. So I'm going to leave it there. I hope I've left some room for Cali. I will take your question at the end.
Okay, thanks, Hossein. So in part 2 of our webinar, we're going to talk about the practice of performing FEA with a focus on the commonly used tools and platforms. At a high level, you can choose between a graphical interface such as web or app, or a statistical programming language. A recent study reviewed the number of newly published tools for FEA over the last 20 years and found 40% were R-based and 34% were web-based. So we'll focus on those two platforms today. Both offer a lot of choice and flexibility, each with their pros and cons. Which suits you depends on a number of factors, including the type of analysis you want to do, whether that be ORA or GSCA, which databases are integrated with the tool and which are most relevant to your particular experiment, the ease of use of the tool, and which species you are studying, because not all tools support all species. The main advantage of web-based platforms is the ease of use with simple point-and-click operations, built-in database integration, and visualization tools. However, they can have limited flexibility, particularly when it comes to visualizing the results. So if the type of graphics offered does not suit your needs, you may need to export those results and replot them. It can also be difficult to obtain details about what is actually going on under the hood, what parameters are applied by default, what are the underlying statistical tests used, and so on. And this can have a major impact on reproducibility. Finally, governance of highly protected data may actually prohibit its analysis online, and that would restrict you to use apps that can be installed locally or to use a programming language such as R. This slide provides an overview of some of the most commonly used web tools and desktop apps for FEA. This list is by no means exhaustive. There are a lot of tools, but it gives an idea of the differing attributes of the various tools, how some of them do many different types of analyses on many different databases, such as string and WebGestalt, while others are more focused, such as Reactome. If you're working with less well-studied species, it's clear that some of the tool choices aren't available to you, and you might need to go with something like gprofile or a string that supports more non-model species. It's important to check what capabilities a tool has to make sure it suits your needs before you decide to use it. When working with web tools, two very common pitfalls have been identified and clearly demonstrated by a recent meta-analysis paper, which you can access via the QR code shown. Firstly, the input of a custom statistical background, or the background gene list that we talked about in part 1 for ORA. Many tools have only recently added this option, and some, such as Reactome, still don't let you upload a custom background. And for those that do, sometimes it's actually quite difficult to find the option to do that. The shown paper found that 95% of the studies reviewed didn't actually include or report an appropriate background. And as we learned in part 1, the inclusion of the background gene list is critical to avoid false positives and bias. Secondly, the black box effect of web platforms can make it hard to find sufficient details to include in your paper for adequate reproducibility. Even with the best intentions, it's so simple to run an analysis, get excited by the follow-up investigation, not save the details of the parameters that you used, and then some months later, when it comes to write the paper, realize that the database version and parameter defaults have changed. Now, this is not a reason to avoid using these tools, but it is a pitfall to keep in mind to ensure that your study does not find itself in one of these unsavory reviews in the future. In contrast, when working with R, your saved code provides thorough reproducibility. There are many dedicated R packages, so you don't need to be coding everything from scratch. And R affords a higher level of flexibility, particularly for plotting. There are many dedicated graphics packages and functions, so you can really tailor the visualizations to suit your needs. The flexibility of R can be very useful if you're working in non-model species or in-house genome assemblies. You can provide annotations of your gene IDs to database terms to perform FEA on any species. And finally, R presents a way of working online, which can be a requirement for highly protected data. The main disadvantage of using R is the steeper learning curve compared to the web platforms. However, there are a lot of online tutorials, shared workflows, and community help pages where someone new to R can obtain plenty of guidance. While R is a command line interface, you can use an integrated development environment or IDE, such as RStudio, and this provides some familiar point-and-click menus, as well as a way to easily manage code, inputs, outputs, and graphics, and create shareable code notebooks all from the one environment. This slide provides an overview of some of the most commonly used R tools for FEA. You'll note that many of these have web counterparts, except Cluster Profiler, which is a dedicated and extremely popular R package for FEA. It has a very comprehensive range of functions and a companion package dedicated to visualizations. Again, note that some tools perform ORA or GSEA or both. Others are more focused on one database, or others integrate with many. Since the inputs required to run these R packages are all the same, your gene lists, the analysis can be done with multiple different R tools in one code notebook. So that can really help if you are wanting to combine your analysis and look for the intersection of significantly enriched terms across different methods as a first step in validating your results. When it comes to choosing your analysis platform, there are three key questions. First of all, is your data highly protected? If so, you might be restricted to using a desktop application or an R programming language. If it's not, and your organism is available on online analysis platforms, or you're comfortable using R, you can have a choice of a number of different platforms. You can do some web analysis and some R. If you're not comfortable with using R, there are options available to you. For example, you can use a string to annotate any proteome if your non-model species is not available on the online web platforms, for example. So it is, there's a lot of flexibility, but those three key questions about your security level of your data, what interface you're most happy to use, and what is supporting your particular species are the key questions in choosing your tools. It's important to note that the choice of tool will impact your results. Statistics is a bit like a Ferrari. Any small change in the input will cause a change in the results. This isn't a problem with the underlying tool. It's due to the fact that all of the tools are doing things slightly differently, different parameters, different databases, and so on. Also, the pre-processing and filtering that you've done to your actual raw data will also impact the results that come out in the enrichment analysis. So this is why recording these details is very critical to reproducibility, because these results are sensitive to all of these factors. As an example of the different input filtering affecting results, here's a gProfiler example on Go terms, the same gene list with different filtering thresholds on fold change and adjusted p-value. You can see that there's a bit of overlap, but there are also some clear differences. So given that all of these filtering options that have been applied to this gene list would be considered reasonable thresholds, which one do we trust? There's no right or wrong answer here, but it does highlight that FEA is a tool to guide your further investigation and hypothesis generation, and not a black and white answer to your study. In this example, the Reactome database was interrogated online with either Reactome or gProfiler using the same input gene list. In both cases, the default analyses were applied, yet you can see some differences in the results. We can also see that there are differences in the actual defaults that were applied by the different tools. So in gProfiler, we can change the FDR method to match Reactomes, but unfortunately we can't upload a custom background gene list in Reactome. So despite getting these two analyses as close as we can to match, there's still going to be differences in the results, and this is expected. Neither one is more right than the other. It just comes down to interpreting the results in line with your particular biological experiment, and then of course performing downstream validation. So understandably, this can be confusing. It's important to keep in mind that as long as you apply robust methods, use sensible parameter choices and perform validation, your results will be as valid as any other, regardless of which platform or tool you use. The authors of Metascape put this nicely. We believe the right attitude on the functional enrichment analysis is to treat it as a guidance to filter and rank pathways and processes, but not to religiously believe in the absolute numbers. So to ensure that you've used robust methods that will stand up to scrutiny and peer review, keep these things in mind. Use the right gene list. So recall from part 1 that there are different input gene lists for ORA and GSEA. If you're doing ORA, ensure to use a custom background gene list to minimize false positives. Always filter on a p-value that has been corrected for multiple testing. Choose actively maintained tools and those that are using the most up-to-date databases. And finally, ensure that you keep a record of all relevant methods so that they can be reported in your paper for utmost reproducibility. In this webinar, we will not go into validation in detail, as this is highly bespoke depending on the nature of your experiment. But a starting point for validate results can be to review the significantly enriched terms, the statistical support that those terms have, whether they are relevant to your experimental condition, or whether they are actually supported when you run the analysis across different tools or different databases. At times, key relevant processes can be obscured by higher order general processes, such as protein binding. As we've seen, the Go terms have a hierarchy. Sometimes you can enhance specificity by using tools such as Revigo or TopGo, which can help group similar terms and isolate key, more specific enrichment results. You can check your workflow against FEA benchmarking tools using gold standard curated data sets, for example, using GSEA benchmark R, but this is a bit of a new and emerging factor in FEA. And finally, in vitro or in vivo validation remains the gold standard form of validation for any results. To wrap up this webinar, let's recap the top 5 things to remember when you're doing FEA. ORA and GSEA are 2 distinct forms of FEA. There are a number of synonyms floating around online for FEA, yet ORA and GSEA are very much not synonyms. They employ different statistical tests, they have different inputs and different biological interpretations. 2, because we are testing hundreds or thousands of genes against thousands of database terms, correcting for multiple testing is critical to avoid false positives. 3, different tools will return different results. This is expected. 4, given the extent to which tool and parameter choice can affect results, thorough method record keeping and reporting is crucial for reproducibility. And finally, remember that FEA is a tool to guide you in understanding your biological system and generating further hypotheses. The FEA results are not the end stage results and do require further critical examination and validation. So that brings us to the end of part 2, and I'll stop sharing and hand back to Melissa.
So I think we'll begin. We have a question here to begin with, and it's what's the minimum number of genes that you need for a functional enrichment analysis?
Okay, so if you're going to choose an overrepresentation analysis or ORA, it's basically the number of genes that have been differentially expressed. You will take them. But what is the minimum number that can satisfy a functional enrichment analysis? It really depends on the statistical methods behind different tools. But if you have noticed, there are some settings like minimum of 15 or 30 genes in some tools to actually test a gene set, for example. So I would say the larger, the better. But on the other hand, you don't want to go too small because it may not be meaningful at all. Let's say if you have 5 differentially expressed genes, it has almost nothing to say. For tests that requires at least 15 to 30 for example, number of genes for a gene set to test. So there's not a clear answer for the number of minimum genes for functional enrichment analysis, but usually for an omics study, we come up with a big number of genes or a list of genes. Yeah, that's not a big deal.
And I guess you can always just plug that gene list into one of the methods and see what the statistic shows you. That's an easy way to see if you've got enough genes there to
see if the statistical method supported or not, correct.
Another question about gene lists. Can these lists also be from differential methylated regions?
Gene list or what does it mean? Gene list or the gene set?
The list that you're from your experiment.
Yeah, I mean, the list of the genes, it can be differentially expressed genes, or it can be a cluster of genes, which is coming out from a WGCNA analysis, or it can be cell type specific. It can be anything that you are interested, really, it means to you in terms of biology, it is meaningful to you. You can use them as an input list of genes.
And we have used the phrase gene list here quite a lot, but this also applies to proteins, for instance. So is that correct?
Yeah, yeah. Well, I've used the term genes, but the right term would be feature, really, because it can be any feature, any biomolecules. It can be genes, proteins, metabolites, lipids, etc.
Excellent, thank you. So let's just go to some other questions here. What are the best practices for carrying out this type of analysis in non-model organisms that may have poorly annotated genomes?
Wow. I'll take this one. Yeah, I've recently come off a project where we had 2 species which were not available on the web tools. So there are 2 options. You can perform an annotation yourself, which of course is, annotation requires a lot of curation to get really good quality results, but there's various tools that you can do that for. And then you can use some mapping. So you map your genes to the databases so that you get the database terms associated with your gene IDs, and then you provide those lists to the tools. And so some tools do allow this, WebGestalt being one of them. And in R, you can have Cluster Profiler or WebGestalt R. If you don't want to do the annotation of your particular genes to database terms yourself, if you don't have the command line ability or access to the right compute, you can actually now do it on string, where you upload your proteome to the string database, and it will create a searchable string database basically for your custom organism, and that's citable, so you can include your link in the publication and you can share it with your collaborators. So even if you are working with a very understudied species, as long as you've got some kind of reference, even if it's not a great reference genome, and you've aligned your expressed data to that reference, that can help with the annotation, improving the quality of the annotation and obtaining your proteome. And from your proteome, you can go ahead and continue to get your annotations against the databases and perform FEA.
Thanks, Cali. Lots of ideas there to get people started. I can see there's a few people in the audience who are working in that area of non-model organisms. A related question is, how can you generate a gene set for specific target genes if they're not available on online databases?
I think if a gene set is not available in database, it means that you are coming up with a set of genes in your experiment. So you're running an experiment in your lab for different conditions, and you will find out with those conditions that you are applying in your lab, you're coming up with the expressions of some genes. It can be in some different time points, or it can be for different, let's say, different concentration of drug, or etc., etc. Those can be regarded as a gene set for you to test in future, I would say.
Thanks, Hossein. A question here as well, which will perhaps need some clarification from you. Is the enrichment analysis related to the length of a gene or protein, and can it be used on small proteins?
Not really directly. We've got these lengths of genes or proteins which can, particularly genes in the gene expression with the past technology in the sequencing, that it can impact the, for example, RNA-Seq experiments, and it can essentially impact the list of genes that you're providing. But these days, technology, it's quite clever, and they are aware of the gene lengths, and there are statistical methods can take care of the gene length variations. So it really depends on the input that you are providing. The functional enrichment analysis doesn't know what you have done beforehand. It's your responsibility that what you are providing for the enrichment analysis.
So the enrichment analysis itself doesn't actually care about the length of the gene or the protein. It just cares about what we know about those genes and proteins in terms of what they do.
Yes, that's right. But if the question is about the length, the size of the gene set, yeah, that's a different thing. But I think this is not the question. The size of the gene set can be an issue, and it can impose some bias in the result. Some of the method can take care of them, but the studies show that can be a source of confusion.
Great, thank you. And one last question to round us out. What's the best background set for a whole organism? For example, if you're doing bulk RNA-Seq analysis.
If you're running bulk RNA-Seq experiment, you would take the entire genes which have been listed in your experiment at the end. Doesn't matter whether they are differentially expressed or not. They have been just some counts for them in your experiment. You just take those as a background, because you're basically telling that these list of genes as a background, they had this chance to express themselves in my experiment, no matter whether they are differentially expressed or not, or they are significant or not. They had this chance to be in the pool, so I take this one as my data, as my background set.
Thank you, Hossein. So we are going to have to leave it there for today. As we mentioned earlier, this webinar is a prelude to a workshop that we'll be running in November, which gives you a chance to get hands-on with some of the web-based and R-based tools for functional enrichment analysis. You can find more information about this workshop on our website. The link for that has just been pasted into the chat. And you can also find out more information about other upcoming events and activities that the Biocommons is doing through our social media by signing up to one of our mailing lists or subscribing to our monthly newsletter, which will be coming out next week. That just leaves me to thank Hossein and Cali again for sharing their time and expertise with us today. I would also like to acknowledge that Australian Biocommons is enabled by ANCRIS funding via Bioplatforms Australia. Thank you to everyone for joining us. We hope you enjoyed the webinar, and we look forward to seeing you again soon. But until then, goodbye for now and enjoy the rest of your day.